This is Andy Alsbaugh with Duke University School of Medicine. In this presentation, we will be discussing the very complex microbiology of pneumonias. So our learning objectives will be first to be aware of the spectrum of pathogens associated with various pneumonia syndromes, and to identify some common bacterial causes and microbiological features of these agents in the following contexts. First, the agents of community-acquired pneumonia syndromes, as well as unique populations at risk for pneumonias, including those with aspiration potential, those with underlying lung disease such as cystic fibrosis or chronic obstructive pulmonary disease, patients with various immunocompromising disorders, as well as pneumonia in hospitalized patients. Going back to our pathogen map, many of these microorganisms can be associated with lung infections. Some of the common bacterial causes are listed here, and we'll discuss them in detail during the course of this talk. As well, several viruses can cause infections of the respiratory tract, especially involving the lungs in a viral pneumonitis syndrome. Additionally, various fungal pathogens enter the body through the host and can cause primary pneumonias at this site. Importantly, pneumocystis, an unusual fungal pathogen, will be discussed in its own separate lecture. Among the bacteria associated with pneumonia, two pathogens are commonly considered very common causes of typical bacterial pneumonia syndromes, including Streptococcus pneumoniae and Haemophilus influenzae. There are other atypical bacterial pathogens, such as Mycoplasma species, Chlamydophila, and Legionella, that collectively comprise the atypical bacterial causes of pneumonia. Many other bacteria can cause pneumonia, including enteric flora associated with hospital-acquired pneumonia syndromes and aspiration, Pseudomonas seen in various obstructive lung diseases, and again, more resistant pathogens seen in hospitalized patients. I alluded to the concept of typical versus atypical pneumonia, and this is an overlapping but historically important distinction that can offer some clinical clues. The typical bacterial pathogens of pneumonia were often seen by gram stain of expectorate sputum and were able to be cultured from this specimen. These would include agents such as Streptococcus pneumoniae and Haemophilus influenzae. In contrast, the atypical bacterial pathogens were not able to be cultured on routine media found in the clinical microbiology lab, nor were they easily identified by gram stain. These would include bacterial species such as Mycoplasma, Legionella, and Chlamydophila, formerly known as chlamydia. Of the typical bacterial pathogens, the most common cause of bacterial pneumonia is Streptococcus pneumoniae, or the pneumococcus. Again, this organism can colonize the oropharyngeal epithelium in many healthy and asymptomatic individuals, and it's only when it reaches the lungs that it's actually able to cause pneumonia. This organism is a gram-positive bacterium, and it has a lancet-shaped diplococcus morphology as demonstrated in the gram stain of an expectorate sputum on the right side of the slide. Like all streptococci, it is catalase negative. On a blood agar plate, it will produce an alpha pattern of hemolysis, and to differentiate it from other similar streptococcal species, it is aptotion sensitive. One of the most important features of Streptococcus pneumoniae is its polysaccharide capsule, and this can account for more than 90 serotypes of this organism. Pathophysiologically, it is the focus of protective immunity. So when one develops immunity against a certain strain, you're protected against reinfection due to that strain based on your reaction to that particular capsule serotype. However, there is another polysaccharide called the C polysaccharide that's present in all serotypes, and it is the basis for a very sensitive and specific urinary test for the diagnosis of invasive disseminated pneumococcal disease. In the past several decades, there's been increasing antibiotic resistance among strep pneumoniae, and this is very important in treatment considerations. Another common cause of typical bacterial pneumonias is Haemophilus influenzae. Similar to strep pneumo, it can colonize the oropharyngeal epithelium in healthy individuals, but in contrast, it is a small gram-negative organism, and it has a shape referred to as a coccobacillus, somewhere between a rod and a round form. Again, similar to Streptococcus pneumoniae, Haemophilus influenza is often encapsulated, and this capsule can be a virulence factor. It prevents phagocytosis as well as lysis by complement. It is also the focus of the protective adaptive immunity, in that antibodies and other cellular features of immunity are directed against this capsule and can protect the host from reinfection. Encapsulated strains are referred to as typable because of the ability to develop antibodies against the various polysaccharide capsule antigens. An example of this would be the well-characterized Haemophilus influenza type B. However, many unencapsulated strains, which are referred to as the untypable Haemophilus influenzae strains, are also frequent causes of pneumonia syndromes. And so someone who would have been vaccinated with the Haemophilus influenza type B vaccine would not be protected from these other unencapsulated strains. Although they are less invasive than some of the encapsulated strains, they can still cause human disease. In terms of the atypical bacterial causes of pneumonia, one of the most common is Mycoplasma pneumoniae. Now, the mycoplasma are the smallest self-replicating organisms that are known, and when they do grow on artificial media, they have this appearance on the colonial appearance as a fried egg, as demonstrated in the right-hand side of this slide. Practically, cultures are rarely performed for this agent, and either the organism is diagnosed by PCR or other emerging molecular tests, or else it is generally just treated by empiric therapy. Microbiologically, this bacterium lacks peptidoglycan. Therefore, it's resistant to many of the cell wall targeting antibiotics, such as the beta-lactams. How does this organism cause disease? Well, first, Mycoplasma species are able to attach to respiratory epithelial cells by important microbiological cellular structures. Once they attach, they're able to enter into these human cells, where they undergo intracellular replication, eventually leading to host cell lysis. Now, again, one of the most important historical features of Mycoplasma pneumoniae is it's associated with what's referred to as a cold agglutinin syndrome. Oftentimes, this infection can induce the production of antibodies that will agglutinate red blood cells in the presence of cold temperatures. This can result sometimes in a hemolytic anemic syndrome, and can also be the basis for the diagnostic test looking for red blood cell agglutination in the cold. This is not specific for Mycoplasma pneumoniae, because other syndromes can also cause cold agglutinin disease. Another type of bacterium that causes an atypical pneumonia syndrome is Chlamydophila pneumonia. These organisms used to be referred to as chlamydia, but the name has recently changed. Again, this is an obligate intracellular bacterium, and if you look at the right-hand side of the slide, you can see the cell labeled as number 1 is a cell that is infected with Chlamydophila species, as demonstrated by the green intracellular inclusions, the intracellularly replicating bacterium, demonstrated by the number 3. Cells that are uninfected are demonstrated by the number 2, and the nucleus is stained, as demonstrated by the number 4. Oftentimes, this organism was referred to as the Taiwan acute respiratory agent, or TWAR. And since it is an intracellular bacterium, it's very difficult to grow and to replicate in culture, so it is infrequently identified during the acute infection, and is usually only identified retrospectively by serology tests. But these serologic tests have indicated a wide range of infections due to this organism and pneumonia syndromes, ranging from anywhere from 1% to 20% of community-acquired pneumonia syndromes. A different species of Chlamydophila, C. psittaci, is a zoonosis associated with bird infections. The third atypical bacterial cause of a pneumonia syndrome is Legionella pneumophila. This is a small gram-negative bacillus that makes beta-lactamases, therefore it's resistant to this class of agents. And it can enter and replicate within macrophages. It's demonstrated on the right-hand side of the slide, where a macrophage is filled with Legionella pneumophila species. This organism is the causative agent of Legionnaire's disease, which historically was described as a fulminant pneumonia syndrome, often associated with contaminated water in air conditioning systems and hotels. Now, when patients come into the hospital with pneumonia syndrome and within 24 or 48 hours end up in the intensive care unit because they have a rapidly progressive pneumonia syndrome, this is one of the organisms that must be considered. Importantly, there is a urinary antigen test that is very sensitive and specific for making the diagnosis of Legionella pneumonia. There are certain predisposing conditions that make one think about different bacterial species other than the ones that I just mentioned. In the context of aspiration, one must consider the contents of the oropharynx entering into the lungs. And depending on the patient population, especially in debilitated patients, this can include enteric gram-negative rods and other resistant gram-negative organisms. Again, oropharyngeal flora could include Staph aureus or other streptococcal species, which might also be implicated in aspiration pneumonias. In patients who have obstructive lung disease, including COPD or cystic fibrosis, the airways can become colonized with various organisms, and notably Pseudomonas and Staph aureus can often colonize patients who have cystic fibrosis, and other gram-negative organisms are often found in the airways of patients with COPD. Immunocompromised patients frequently develop pneumonia, as this organ is in constant communication with the outside world. And many different organisms can cause pneumonia syndromes. But you must remember the typical and atypical causes of community-acquired pneumonia in these patient populations. And as well, there are certain fungi that can cause lung infections in highly immunocompromised patients, including Pneumocystis species, the mold Aspergillus, as well as the yeast Cryptococcus and the dimorphic fungus Histoplasma. Various respiratory viruses, including community-acquired respiratory viruses, but also opportunistic respiratory viruses such as cytomegalovirus, can cause lung infections in immunocompromised hosts. And there are also protozoal and nematodes that can cause lung infections, including Strongyloides, which can cause a lung and hyperinfection syndrome in immunocompromised patients. Hospitalized patients represent a specialized and important group of patients who can develop pneumonia. The microbiota of hospitalized patients undergoes predictable alterations that put them at risk for different pathogens that must be considered, including some multi-drug resistant gram-negative rods. Mechanical factors, including endotracheal tubes and patient debility resulting in increased risk for aspiration, as well as immunocompromising disorders, also make hospitalized patients at high risk for consideration of different organisms than those in patients developing pneumonia from the community. So here's a list of other organisms to think about in pneumonia syndromes in hospitalized patients. This chart summarizes the patient and microbial factors to consider in individual patients discussed in the previous slides. Now, also, the etiology of pneumonia can be distinguished by patient age. So in neonates, they're often exposed to group B Streptococcus and E. coli during the birthing process. So these are agents that must be considered in any kind of neonatal infectious syndrome. RSV can cause a particularly devastating form of pneumonitis in this age group as well. As babies get a little bit older, again, Streptococcus pneumoniae and chlamydia species can cause pneumonias more frequently in this population, and other community-acquired viral pathogens are also very important to consider within young infants. As the immune system begins to mature and the microbiota changes somewhat, children become more susceptible to the common bacterial causes of pneumonia, including strep pneumoniae, Haemophilus influenzae, and other community-acquired respiratory viruses. Young adults can often have walking pneumonia syndromes due to the atypical bacterial species, as well as Streptococcus pneumoniae, as well as community-acquired respiratory viruses. Older adults, Haemophilus influenza tends to be present at a bit more frequency, and they are at high risk of influenza, making influenza vaccination a very important preventive strategy in this patient group.